Welcome student for today's lecture. Student, today we are going to see the next point, that is about the translation. That is in the protein synthesis process, that is there are the two steps. One is the transcription and another is the translation. So student, today we are going to see the next point, that is about the translation. So translation, during which process here actual protein are synthesized. So how the protein synthesis takes place, that is here the codons of the mRNA, it get translated. These codons which get translated in the cytoplasm of the cell, and when it get translate, it produce the polypeptide chain of the amino acids. Polypeptide amino acid ची polypeptide chain तयार होईल. It is nothing but the proteins. So this process is the translation. So what happens, that is here during the process of translation, that is the protein synthesis takes place, that is there is the formation of polypeptide chain of the amino acids. So first upon see what are the requirements, basic requirements for the translation process. So the basic requirements, these are first, that is the amino acids. So amino acids, these are mainly 20 amino acids which are required for the protein synthesis, and in the cell there are the 20 amino acids are. Then RNA, that is there are the three types of the RNA, mRNA, rRNA and the tRNA. mRNA, that is the messenger RNA, rRNA ribosomal RNA, and tRNA, that is the transfer RNA. These three RNA are required. Then third, that is the ribosomes. RNA, then ribosomes. So ribosomes, these are in the eukaryotic cell and the prokaryotic cell. That is in the prokaryotic cell there is the 70S type of the ribosome, while in eukaryotic cell that is the 80S type of the ribosomes are required. Then ATP, it is the energy which is required for the bond formation. Then magnesium ions are also required. Then enzymes, then the processes, that is the elongation, then translocation and the release factor. This release factor, it mainly help to release the polypeptide chain, that is when the ribosomes reach at the 3' end or at the termination codons. Okay. So these are the requirements for the protein synthesis, that is the process of the translation. So see, that is here for the translation process, these are some requirements, out of which here the ribosomes, these are most important which are required for the process. And first we see, that is about the structure of the ribosomes. So see, that is here the process, that is the translation process, it mainly takes place, that is in the cell cytoplasm. So here in the cell cytoplasm, all the requirements are there. So ribosomes, so first we see, that is about the ribosome, that is how the ribosomes are. So ribosomes, if we see, that is in the ribosomes, they are freely suspended in the cell cytoplasm. It is having the two units, that is the larger subunit and the smaller subunit. So if we see the structure of the ribosome, that is the ribosome, it is having the larger subunit. These ribosomes, these are mainly freely suspended, that is in the cell cytoplasm. So larger subunit, and there is the smaller subunit. So see, that is here the ribosomes, which is having the larger subunit, it mainly sac-like structure, and it is mainly having the components, that is the proteins, then rRNA, that is the ribosomal RNA. Then it is also having the magnesium ions. Then in the ribosomes also the calcium ions are also present, that is in the larger subunit. So the ribosome having the larger subunit, smaller subunit. In the larger subunit, that is there is also presence of the calcium ions. So proteins, rRNA, magnesium, calcium ions are present in the larger subunit of the uh, ribosomes. So these both subunits, they are freely suspended in the cell cytoplasm, and during the protein synthesis they attach together on the mRNA. So see, that is here the in the eukaryotic cell, if we see, that is in case of the eukaryotic cell, that is this ribosome is of the 80s, that is it mainly having the two subunit, that is the 60s and the 40s. So see, that is here the in the eukaryotic cell the ribosome, it is of the 80s, that is equal to 60s plus 40s. That is here the two subunits. So the larger subunit, it is of the 60s, while here the smaller subunit, it is of the 40s. In the eukaryotic cell, that is here it is mainly made up of the 80s. So 80s, that is having the 60s, that is the larger subunit, and 40s is the smaller subunit. If we see in the prokaryotic cell, that is in the prokaryotic cell, it mainly having the 70s type of the in prokaryotes. It is mainly 70s, that is out of which here the larger is 50s plus a smaller is of the 30s. So here the larger subunit in the prokaryotic cell, if we see, that is the larger subunit is of the 50s and the smaller subunit is of the 30s. So that is 50s and the 30s, when combines together, it produces the 70s, that is the two subunits, that is S, that is means that is the Swedberg constant. S म्हणजे Swedberg constant असं म्हटलं जातं. तर इथं युकॅरोटिक सेलमध्ये जनरली आपण पाहिलं तर रायबोसोम हा 80s असतो, तर प्रोकॅरोटिक सेलमध्ये पाहिलं तर तो किती असतो 70s, दॅट इज द about the ribosome. So next, that is how the structure of the ribosome, that is during the protein synthesis, that is during the polypeptide chain formation, the two subunits of the ribosomes, they are combined together or bind, and here the there are binding site for the smaller subunit and the larger subunit. So see, that is here, see here the larger subunit, if we see, so having the structure like this. So here the larger subunit, and this it is having, there is the smaller subunit like this. So these two units gets bind during the protein synthesis. So larger subunit, it mainly shows the three sites like this. So one, that is the, it is the E site, then it is this site is the P site, and this site is the A site. So these are the three sites at the larger subunit. In case of the eukaryotic cell, the larger subunit, it is mainly of the 60S, while the smaller subunit, it is of the 40S. Okay, so in the larger subunit, that is here, see, that is there are the three sites. So E site means the exit site. E site is called as the exit site. So through this exit site, here the tRNA, that is it mainly discharged, that is the empty tRNA is released. Through the E site, empty tRNA is released. Then P site, it is the peptidyl site. P site is the peptidyl site. So through this peptidyl site or P site here, the polypeptide chain is released. And A site, that is, it is the acceptor site. Amino acyl tRNA complex is mainly enters through this A site. So A site is the acceptor site. A site is the acceptor site. Through this acceptor site here, the amino acyl tRNA complex, it mainly enters into the ribosomes. Okay, so it is the structure of the ribosome, and here today we see, that is next part, that is how the translation process takes place. Okay, so it is about the structure of the ribosome. In the larger subunit, there are the three sites, E site, P site and the A site. The three steps, first that is the initiation, second is the elongation and third is the termination of polypeptide chain. So here the termination of the polypeptide chain. So first upon we see the first step, that is the initiation. So initiation means here there is the first step of the initiation, that is activation of the tRNA, that is the transfer RNA takes place. So what is mean by the activation of the transfer RNA? That is here the tRNA gets charged. So how the charging of the tRNA takes place? That is here in the cell cytosol, that is there is the amino acid pool. There are the 20 amino acids, and here in presence of ATP, that is, and the amino acyl tRNA synthetase enzyme, the tRNA, that is transfer RNA, gets charged. That is here the tRNA having the two ends. Here is the 5' end and here is the 3' end. So at the 3' end there is the longest end, that is having the CCA end, and at this end here the amino acid gets attached. So here this amino acid gets attached at the 3' end of the tRNA, that is at the CCA end. Okay, and here the tRNA, amino acyl tRNA complex, which is produced. So the structure is known as the amino acyl tRNA complex, or it is also known as the charged tRNA. It is also known as the charged tRNA. So here the during the initiation, म्हणजे पहिली सुरुवात, ही पॉलिपेप्टाइड चेन तयार होण्याची पहिली सुरुवात म्हणजे इनिशिएशन, आणि इनिशिएशन होताना पहिली स्टेप होते, दॅट इज द ॲक्टिवेशन ऑफ द tRNA. आणि tRNA ॲक्टिवेट होतो म्हणजे काय होतं? इन प्रेझेन्स ऑफ द ATP अँड हियर अमायनो असाइल tRNA सिंथेटेज कॉम्प्लेक्स एन्झाईम, हियर द amino acyl tRNA complex is produced. Okay, so here it is known as the charged tRNA, that is the amino acid gets attached at the 3' end, that is the longest end of the tRNA. So here the amino acyl tRNA complex is produced. Then next, that is here the two subunits of the ribosomes, that is they gets attached to the mRNA. So this structure is the mRNA. It is the mRNA. So how the mRNA is? That is mRNA, it is having the 5 prime end and the 3 prime end. At the 5 prime end there are the initiation codon, 3 prime end there are the termination codon. So on this mRNA there is a specific region it is produced, that is there are the codons, and this region it is known as the UTR region, that is the untranslated region. So this untranslated region or the UTR region, it mainly present before the initiation codon. So see, that is here are the initiation codon, before which here is the untranslated region is present. So before the initiation codon, that is at the 5 prime end. Then after the termination codon, UAA, UGA and UAG, these are the termination codon. So after this termination codon, this region, at this region, here is the UTR region, and this is the untranslated region of the mRNA. So this mRNA having the untranslated region, that is untranslated region are the most important for the process of the translation, and the mRNA without the untranslated region, or which mRNA which are not having the untranslated region or the UTR region, they are unable to translate, that is this mRNA which are not having the UTR region, they does not produce, this mRNA does not produce the proteins. Okay. So the most important region, that is the UTR region. Then next that is about the ribosomes. So the ribosomes having the larger subunit, we have seen that is there are the 3 sites, that is the E site, P site and the A site. So E site is the exit site, P site peptidyl site, A site is the acceptor site. So here the first upon here the smaller subunit of the ribosome is attached at the UTR region. म्हणजे हा रायबोसोमचा स्मॉलर सब युनिट जो आहे तो सुरुवातीला या UTR regionला अटॅच होईल, then the larger subunit gets attached. So see, that is here when the larger subunit and the smaller subunit get attached on the mRNA, then what happens, that is during the initiation, so first step takes place, that is the charged tRNA, it enters through the P site, that is here the P site. See here there is the P site. This P site directly accept the charged tRNA. So see, that is here it is the charged tRNA. So here is the amino acid first. Okay, so here this tRNA, it mainly first tRNA, which mainly codes the initiation codon, that is the AUG, and this tRNA, the first tRNA only enters through the P site, but the other tRNA, that is which are codes by the codons, this tRNA it mainly enters through the A site. Only the first tRNA, that is the tRNA complex or the charged tRNA, enters only the P site. So see, that is here in the eukaryotic cell, the charged tRNA, it mainly carried the amino acid, that is the methionine. So in the eukaryotic cell, here the amino acid which is carried, that is the methionine, that is the methionine, the first amino acid, it is carried by in the eukaryotic cell. So here this tRNA, that is first tRNA, it enters through the, remember लक्षात ठेवा की पहिला tRNA जेव्हा या रायबोसोम मध्ये येईल तो tRNA हा नेहमी P साइट मधून येईल, बाकीचे राहिलेले जेव्हा tRNA एंटर होतील ते मात्र ॲक्सेप्टर साइट म्हणजे A साइट मधून एंटर होतील. तर याच्यामध्ये यूकॅरियोटिक सेल जर असेल तर इथं मिथिओनाइन हा अमायनो ॲसिड carry केला जाईल by the tRNA. While here in the prokaryotic cell it is the formyl methionine. म्हणजे प्रोकॅरियोटिक सेल मध्ये जर पाहिलं आपण तर या फॉर्मिल methionine. It is carried by the tRNA. म्हणजे प्रोकॅरियोटिक सेलमध्ये जेव्हा प्रोटीन सिंथेसिस होईल तेव्हा पहिला अमायनो ॲसिड जो येईल तो फॉर्मिल मिथिओनाइन. आणि जर युकॅरियोटिक सेल असेल तर पहिला अमायनो ॲसिड जो tRNA कॅरी करेल दॅट इज द मिथिओनाइन. ओके, सो द फर्स्ट स्टेप दॅट इज इट इज द इनिशिएशन. मग इनिशिएशनमध्ये पहिली स्टेपमध्ये काय होईल, जर इथं AUG हे जर तीन इनिशिएशन कोडॉन असतील तर याला कॉम्प्लिमेंटरी अँटीकोडॉन्स हे कुठे असतील, या tRNAवरती असतील. म्हणजे इथं ॲडिनाइन आहे तर इथं असेल युरॅसिल, इथं युरॅसिल आहे तर इथं असेल ॲडिनाइन, आणि इथं गॉनिन आहे तर इथं कोणतं असेल, सायटोसिन. सो हियर द फर्स्ट tRNA that is enters through this P site that is having the anticodons that is the UAC. And in the eukaryotic cell this tRNA carries the amino acid that is the methionine, while in the prokaryotic cell the amino acid it is carried that is the formyl methionine. Okay, so it is about the first step that is the initiation.